for our final speaker today, it's a privilege to introduce Dr. Maria Letizia Barreca from the Department of Pharmaceutical Sciences at the University of Perugia in Italy. She is a medicinal chemist, which is, she thinks she might be the only one in the room, and she's probably right.
It's a rare species, I think, here.
Okay. Anyways, Dr. Barreca is going to be talking about her grant entitled Blocking the Neurotoxic Activities of Mutant Prion Proteins by a Novel Class of Therapeutic Agents. Please.
Thank you for your kind presentation. Yeah, it's a great honor for me to be here today. And first of all, I really would like to thank the donors for their support, because without their help, I couldn't afford some of the experiments that I'm going to present today. And yesterday evening, I had the opportunity to meet one family, and it was a great emotion to me. So thanks a lot. And thanks also to the CGD Foundation for their great work in this field. So our project is about drug discovery. Drug discovery is a very long and expensive journey, and the success rate is very low, and you need to put together different expertise and competence to hopefully reach the goal. And this is our case. So we have a team, all Italian people, but they are working in different parts of Italy, and we have different skills. At University of Perugia, we are a medicinal chemistry team, and I and my colleague, Giuseppe Manfroni, so we are the person that are thinking about the compounds, the molecules that can be tested in some biological testing. And then we have two experts, of course, of prion diseases, and these are Emiliano Biasini and Romolo Nonno. And Emiliano Biasini was also a recipient of other CGD Foundation grants in the past. And when you are talking about drug discovery, you also need a pharmacologist in the team, and our expert is Francesca Fallarino. So before going into the detail of the research activities that we have performed using CGD Foundation funding, I have to give you a brief overview of what we have done before. So it's well established that, the key event for the development of prion disease, you heard many times, in these days, is the conversion of the Prpc, the physiological form, in the pathological form, the Prpsc, that is able to aggregate and accumulate in the brain and damage it. But there is a key role in this process given by the Prpc, by the physiological protein. Indeed, as you know, he is able to be the substrate for the conversion of the Prpsc in multiple amounts of pathological form. This is just a model. So Prpsc is getting in touch with the physiological protein, and then the Prpc is converted in Prpsc, and then you have a large amount of the pathological form. But the Prpc is doing something else. So there is, different evidence that Prpc is also important for neurotoxicity. As you heard, this kind of mechanism is not well understood yet. There are different hypotheses. And in one of these hypotheses, and again, this is a model, you have that Prpsc is getting in touch with Prpc, but then something happens and you have neurotoxicity. So, and this particular, mechanism was really important to us because we decided to work on a drug discovery project that was able, hopefully, to identify compounds able to block this toxicity that was mediated by PrP. How to identify these compounds, which is the starting point. So we used a quick cellular assay that is called DBCA. So this assay was originally developed at the lab of Dead Virus in Boston. And at that time, Emiliano Biasini was a postdoc in this lab. And you can think about this cellular model as a surrogate model to study, to identify compounds that have some potential to block neurotoxicity. But this is just a primary screening, It's just able to select compound that can have some potential. But later you need to perform additional experiments to be sure about your results. And the DBCA was already used to screen compounds, and among the molecules that were published, we chose to work on this specific compound. because, I mean, the structure of these compounds was known on our team in Perugia, because we were already working on other compounds that were similar to this one, even though for other diseases. So this was the starting point, and in our lab, we started to think how to modify these compounds, so the design part, and then to synthesize, that means to prepare the compounds. And we prepared about 80 compounds. And then I can tell you that for an academic group, this is a huge number. And then the compounds were tested in the DBCA. And we were able to find some compounds that were more potent of the parent compound. Just to better understand that parameter, RD50, is just a measure of the potency of one compound in DBCA. The lower the value, the more potent is the compound. So we were able to find this, first compound, and as I said, DBCA is a primary screening, so we need to, perform other additional experiments. So we saw that this compound was not able to target directly, the PrP. So the PrP was not the target of our compounds. In other words, it was not able to bind to Prpc. And also our compound was not able, to block the replication of Prpsc. So he didn't act on propagation. But this was not surprising to us because, remember, I said that we wanted to work on neurotoxicity and not on replication. And then also we saw that, we observed that our compound was also able, to suppress some abnormal electrophysiological activity that was induced by mutant toxic PrP molecules. So everything, was promising here, but now is the bad news. The bad news is that we did some additional experiments, and we saw that, unfortunately, our compounds were not suitable for in vivo studies, animal models of prion disease. And this because the compounds, was not stable. This means that if our compounds was provided to an organism, cells or an animal models on humans, it was destroyed immediately. So it was not able to perform any kind of activity. So again, we went back to the medicinal chemist lab and we started to design and synthesize more compounds. And we were able, to have a pool of new compounds that already were tested on DBCA and other experiments. And we were able to get, even more potent compounds. You remember? RD50, lower, more potent. And also they were not toxic in cells. What does it mean? This means that it's not toxic in normal cell, because of course you want to kill the cells that have a disease. You don't want to kill the normal cells. And at this point, before going ahead, we did an additional experiment to have an additional proof that our compounds could work on toxicity. So this is an ex vivo model from prion toxicity, and what we have seen is that our molecule was able to restore normal synaptic functionality, at least in this test. And here we come with the CJD Foundation. So we had this background, and now we said, okay, maybe we could be ready to have additional experiments, to go to in vivo models. And first of all, we need to, synthesize a great amounts of compounds. The experiments that I said before in vitro just required a small amount of compounds, like milligrams. But if you have to go to the animal models, you need grams of compounds. So we could, pay a person that was dedicated to this work. You see how many steps to generate, just one compound. And this person was dedicated, Maria Giulia. She was already a PhD student in our lab at Perugia, and she was working hard to get the good amount for the compounds for additional experiments. And then the big question was, okay, the compound doesn't work on PrP, but which is the mechanism of action, which is the target. And this was really important to us before going in vivo models, because first of all, you need to know how my compound works. And the second is that if you know the mechanism of action, you can also have an idea about the side effects that you can have in vivo. So for this, particular, work, we didn't have the expertise, so we did this in collaboration with the EU Open Screen Drive. They did a big work, trying to, the kind of what is a target fishing, so to try to identify the target. And we have to synthesize some particular compound. And then finally, they provide us a short list of targets that we are going to validate. And then, for the in vivo studies in animal models of the disease, you need before to do some additional experiment, both in cells and in another kind of animal models, to study what is called ADME and the pharmacokinetic profile. This means that you want to know what happens to my molecule when I put in my compound within an organism before reaching, the target. This means that is the molecule stable, or will be the molecule be destroyed in the organism? Is the molecule able to cross the brain membrane? How much compound can we have within the brain? So this experiment is very important because before going in, in mouse models or animal models of diseases, because you need to collect information that you can use then to set up your trial in vivo. And for the in vitro part, we have already the results, and these are very promising, and this was the more expensive part of this project, and we performed this with a partner, an external partner, this Alvita, and the in vivo part is also over, so it's finished, but we still have to collect, the final report, and we are keeping cross our fingers, because if, the results will be promising, we will go to the animal models. So, what I can do to summarize. So we have a new chemical class of compounds that show some promise. I cannot say that we have, a drug, but we have some promise. We are working hard to going ahead in the drug discovery pipeline. And of course, we are still working much efforts to validate the target. So we know now which might be the targets. We have already, some biophysical studies ongoing to see if our compound is able to bind any of this protein. And we have also to analyze the in vivo experiment, as I said before, in order to set up the in vivo studies on the animal models. And we would like really to have a proof that these molecules can give us some kind of benefits, in this devastating disease. But we have also to remember, as I said before, that there is still a lot of work to do to understand prion biology, especially neurotoxicity. So these compounds, maybe there will be some drug candidate in future, but they can also serve as chemical tool, chemical biological tool, to try to better understand some understood aspects of prion biology as neurotoxicity. So again, thank you all for your attention, and again, thanks to the donors and to the CGD Foundation for their great support.